So a pleasant morning, Ma'am Sawi, and to my dearest classmates. We are the last presenter to conduct focus group discussion at itutuloy namin yung nasimulan ng Group 5. So we will be discussing the DNA structure and the central dogma part 2. And of course, at the end of this lesson, all of us should be able to first explain the central dogma, second, explain the main step of transcription. Third, describe how eukaryotic mRNA is processed. Fourth, describe the different steps in protein synthesis. And lastly, discuss the role of ribosomes in protein synthesis. So to formally begin this session, I will call on Ma'am Miriam to take her part.
So, ayan, buhay po ba tayong lahat? A pleasant afternoon, Professor Sawi, and to all of you, classmates. So, I am the first discussant in our group, and my topic is all about the central dogma of molecular biology. Next po. Yan, I'm sure naman lahat kayo is may stock knowledge naman about central dogma. So, first, unang-una pa sa first, may tanong agad. Can you guys give me one word that you could associate with our topic, the central dogma? Any one word po na para sa'yo is related sa central dogma. You can type your answers po in our in-call messages. Thank you po, Ma'am Melissa, Miss Eunicia, RNA, translation, transcription, translation. Okay, thank you po. So, ayan. Thank you, Triby, for all of your answers. They are all indeed connected to our topic. Next po. Ayan, so done na tayo dyan. So, the central dogma is the process by which information flows in living cells. So, paano nga ba? So, the instructions in DNA are converted into a functional protein product. Next po. Ayan. The central dogma of biology explains that the flow of genetic information is from DNA, ayan, sa unang-una, DNA to RNA, making a functional product as a protein. So, next po. So, this idea was first proposed in 1957 by Francis Crick, shortly after nilang i-discover ni James Watson yung structure ng DNA. Since sila yung nakadiscover nun, after nun na pinropose naman ni Francis Crick yung about sa statement niya dito sa central dogma. So, he stated that, wait lang po, pabalik po. DNA makes RNA and RNA makes protein. And he also restated it in a Nature paper published in 1970, saying that the central dogma of molecular biology deals with the detailed residue by residue transfer of sequential information, and it states that such information cannot be transferred back. Ayun, so hindi na daw pwedeng ibalik from protein to either protein or nucleic acid. Central dogma states that once information has passed into a protein, it cannot get out again. So in more detail, the transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein, pwede pa, possible pa. But transfer from protein to protein or from protein to nucleic acid is impossible na. So, information here means that the precise determination of sequence, either of bases in the nucleic acid or of amino acid residues in the protein. So, next po. Ngayon, pag-usapan pa natin ng konti pa yung sinabi ni Francis Crick about the central dogma. So, in the first bullet, in 1957, Crick presented a paper to the Symposium of the Society for Experimental Biology. So, yung title nun is On Protein Synthesis, in which he outlines the base structure of the central dogma. So, doon sa paper na yon, bumuo siya na argument sinabi niya doon na DNA daw is the material responsible for heredity, and as such, DNA is must code. DNA must code for all the molecules needed for life. At doon sa paper na yon, Crick questioned how DNA, which is located inside of the nucleus, could cause protein synthesis, which is alam naman natin na nangyayari sa loob ng nucleus in the cytoplasm. So in the third bullet, Crick postulated that there must be some intermediate molecule that delivers the information from the DNA to the cytoplasm. So sabi niya, hindi naman makakarating yung information kung wala nagdadala nun. So specifically, yun daw yung RNA. And then, dahil dito nabuo niya din yung idea na tinatawag niyang linearity. So, ayan na pala. Linearity in the central dogma. So, states that DNA information is transferred to RNA, yan sa last, which is translated into action in protein synthesis, explaining that once the genetic information reaches the protein form, it can never be reverted back to RNA or DNA. Next slide po. Yan. The central dogma suggests that in living cells, DNA contains the information needed to make all of the cells proteins, and that RNA naman is the messenger that carries these informations to the ribosomes. Next po. So, pwede nating isipin na yung cell is our protein-producing factory. So, yan, tandaan natin yan. Cell is our protein-producing factory, and ribosome naman is our specific protein-building department. Next slide po. So, ang tawag naman sa process ng which DNA instructions are converted into a functional protein product is called gene expression. So, an example, eye color is due to the protein pigment. Next po. Yan. Gene expression has two key stages, which is the transcription and translation. Next po. Ayan. So, transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA or mRNA. Simply put, it is the process by which DNA is copied to RNA, diba? Where the information in the DNA is converted into a small portable RNA messenger called messenger RNA or mRNA. Next po, yan. So, during translation naman, ayan, sabi dyan, it is the process by which a cell makes proteins using the genetic information carried in mRNA. So, during translation, these mRNA messages travel from inside the nucleus, palabas ng ribosomes, in the cytoplasm, kung saan doon sila binabasa to make a specific protein. Next po. Ayun. So, the central dogma states that the pattern of information that occurs most frequently in cells is from existing DNA to new DNA, which is called DNA replication. Next po. Yan. DNA replication. Ayun. So, from DNA naman to make new RNA is called transcription. And from RNA to make new proteins, which is called translation. So, meron tayong tinatawag na reverse transcription. Yun naman, ang process nun is the transfer of information from RNA to make new DNA. Yan. So, paano ba yan nangyayari? So, this occurs in the cases of retroviruses such as HIV. Yan. It is the process by which genetic information from RNA is assembled into new DNA. Yan pa, next po. So there's a question here. Does the central dogma always apply? Yes or no po? Sa tingin nyo lang, opinionated. What do you think? Does central dogma always apply ba? Yan, sabi ni Ma'am Suwal, yes. Ma'am Dalatre, yes din daw. Yan, almost all of the answers are yes. So actually, thank you, 3B. So actually, according to the video I watched, with modern research daw, it's becoming that some aspects of central dogma are not entirely accurate in the current researches because there are, in current researches, there are investigations that the function of non-coding RNA, although this does not follow the central dogma, it still has a functional role in the cell. So next po. Yan. In summary, the central dogma of biology is the unifying theme that we see in all living organisms. Yan. Where basically DNA makes RNA and RNA makes protein. Yan, so, but wait, there's more. Tingnan natin kung naintindihan nyo yun, yung maikling discussion na yon. So, I have prepared brain break 9 questions. So, here's the first one. You can answer po your, you can type your answers po in our in-call messages. So, number 1. It is the process by which the DNA instructions are converted into a functional protein product. So, anong tawag daw po doon? Thank you. Gene expression. Tingnan po natin kung tama. Yon, correct. So, next. True or false naman? DNA contains the information needed to make all of the cell's proteins. Is it true or false? Thank you, ang bibilis. Ang tatalino. Yan. Okay, reveal na natin yung answer. It is true. Next. True or false ulit? RNA is the messenger that carries this information to the ribosome. So, mRNA daw ba yung nagkikarry ng information? Ang bilis talaga ni Ma'am Lontayaw. Okay, thank you Ma'am Dalatre. Mga Dean's Lister, tingnan natin kung tama yung sagot nila. Yon, true. Thank you. Next. It is called the specific protein building department. Ayan, yung in-emphasis ko kanina. Ribosome. Thank you guys. So, tingnan natin. Correct. Next po. Yan. It is the protein producing factory. Ano naman yun? Yan, di ba? Basic na basic. Cell. Okay. Next. Ayan, tama. To, it is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. Ano naman daw po yun? Transcription. Thank you guys. So, tingnan natin. Yon, correct. Next po. It is the process by which a cell makes proteins using the genetic information carried in messenger RNA or mRNA. So, ano kaya yon? Kung transcription yung kanina, ayun, translation. Tingnan natin. Yan, tama ulit. Ayaw nyo ba magkamali? Charot. Basic sa inyo na yan eh. So, it is the process by which information flows in living cells. Alaman daw yan. Correct. Central dogma. Ang bilis talaga ni Ma'am Dolatre. Okay. Last na po. Who stated that DNA makes RNA and RNA makes protein? Ayun. Francis Crick, thank you Ma'am Dolatre, Lizander, Regacio, Estrella Lantayo, and company. So, that's all for my part. Thank you po. For the next presenter.
So, good afternoon, everyone. I am Maria Eric as well, and the objective that was given to me is to explain the main steps of transcription. The next po. But before we go further in deeper about transcription and its processes, I have prepared here words that we will encounter in my entire discussion para hindi na kayo mahirapan at hindi na rin ako mahirapan. So, the first word is mRNA. Messenger RNA, a form of RNA that carries the nucleotide sequence code for a protein sequence that is translated into polypeptide sequence. Next, non-template strand. The strand of DNA that is not used to transcribe mRNA. This strand is identical to the mRNA except that the T nucleotides in the DNA are replaced by U nucleotides in the mRNA. The next is promoter, a sequence on DNA to which RNA polymerase and associated factors bind and initiate transcription. Usually, it is located upstream of the transcription unit. Next one is RNA polymerase, an enzyme that synthesizes an RNA strand from a DNA template strand. The next one is template strand, the strand of DNA that specifies the complementary mRNA molecule. The next one is terminator. Terminator is a section of nucleic acid sequence that marks the end of a gene or operon in genomic DNA during transcription. And the last one is transcription bubble, the region of locally unwound DNA that allows for a transcription of mRNA. Next po. So first, I just want to ask, what comes to your mind when you hear the word transcription? So kindly po type your answer pa sa Intel messages. What comes into your mind po when we hear the word transcription? So nababasa ko, galing kay Miss Villaflor. DNA will be converted to mRNA since light po. So, any other answer po? Okay. Yan. So, sige. Okay, transcription, pag naririnig natin, di ba, pumapasok sa isip natin is a written or printed representation of something. So, next question I have is, have you ever had to transcribe something? Na-try niyo na po ba mag-transcribe? For example, you call your mama. Diba? Tumawag kayo sa mama niya. Tapos nag-ask kayo ng information. Diba? Na, kunyari sinasabi na, mapengin ako number ni Papa or any other information. So, syempre, pag sinabi yun ng mama mo, hindi mo naman yan magigets in instant. So, hindi mo siya matatandaan. I-write down mo siya, diba? So, madalas sa chismis. Okay. So, next one naman is, na example is, maybe we took down notes sa klase, then we rewrote them neatly to help us review. Sinong ganon? Madalian muna, tapos uulitin na lang. Pinahirapan yung sarili. So, isa ka doon. So, this example shows transcription. Transcription is a process in which information is rewritten. And transcription is something we do in our daily lives. Minsan hindi lang natin siya napapansin. So, next po. Yan. So, for the short description of transcription, meron tayo sa screen. Transcription is a process of copying a segment of DNA into RNA. The segments of DNA transcribed into RNA molecules that can encode proteins are said to produce messenger RNA. Take note that transcription occurs in the nucleus of the cell and the mRNA transcript must be transported to the cytoplasm. So, transcription is the first step of gene expression. During this process, the DNA sequence of the gene is copied to RNA. Before transcription can take place, the DNA double helix must unwind near the gene that is getting transcribed. The region open up DNA is called transcription bubble. So, yung mamaya yan, mapapakita natin yan kung ano ba yung transcription bubble, kung ano ba yung itsura niyan. So, transcription uses one of the two exposed DNA rather strand as a template. This strand is called the template strand. The RNA product is complementary to the template strand and is almost identical to the other DNA strand, called the non-template or coding strand. However, there is one important difference in the newly made RNA. All of the nucleotides are replaced with the U nucleotide. So, tatandaan niyo sa transcription, yung T-nucleotides natin or yung thiamine is nare-replace na siya ng uracil. So, the site on the DNA from which the first RNA nucleotide is transcribed is called the positive 1 site or 1 site. Or pwede rin siyang tawaging initiation site para mas madali. Nucleotides that comes before the initiation sites are given negative numbers and said to be upstream. Nucleotides that come after the initiation site are marked with positive numbers and said to be downstream. So, next po. So, before we discuss the main steps of transcription, let us know first what is the RNA polymerase. So, the main enzyme involved in transcription is the RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Specifically, RNA polymerase builds an RNA strand in the 5' to 3' direction, adding each new nucleotide to the 3 end of the strand. Pero ano nga ba yung 5' and 3'? At the 5' end of the chain, the phosphate group of the first nucleotide in the chain sticks out. The phosphate group is attached to the 5' carbon of the sugar ring, which is why this is called the 5' end. At the other end, called the 3' end. The hydroxyl of the last nucleotide added to the chain is exposed. The hydroxyl group is attached to the 3' carbon of the sugar ring, which is why this is called the 3' end. So, next po. So, the transcription occurs in 3 main steps. So, the first one is, kindly raise your hand to decode this gibberish po. Sinong gustong sumagot? Hindi ako magna-next pag walang sumagot. Sinasabi ko sa inyo. So, nakita ko si si Miss Kristen Estrella. Pa-open po ng mic.
Initiation po.
Ulit po.
Initiation po.
Okay. Patingnan po natin kung tama po ba. So, tama. May tama ka. So, yan. Thank you sa mga sumasagot. So, next po. So, the first step is initiation. It is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called promoter. This signals the DNA to unwind the enzyme can read the base in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with complementary sequence of bases. So, to begin transcribing a gene, RNA polymerase binds to DNA of the gene at a region called the promoter. Basically, the promoter tells the polymerase where to sit down on the DNA and begin transcribing. Each gene or in bacteria, each group of genes transcribed together has its own promoter. A promoter contains DNA sequence that let RNA polymerase or its helper protein to attach the DNA. Once transcription bubble has formed, the polymerase can start transcribing. So, yung pag nakita niyo yung parang may umbok na bilog, pa-oblong, yan yung transcription bubble. So, transcription requires the DNA double helix to partially unwind in the region of mRNA synthesis. The region of unwinding is called transcription bubble. The DNA sequence onto which the proteins and enzyme involved in transcription bind to initiate the process is called promoter. In most cases, promoters exist upstream of the genes they regulate. The specific sequence of a promoter is very important because it determines whether the corresponding gene is transcribed all of the time, some of the time, or hardly at all. Next po. So, sino ulit ang magde-decode? Kindly type ako na lang. Ayan. Si Miss Emily, mabalad. Nauna siya. Ako na lang. Okay. Okay. Ikaw na. Kindly open your... Hindi po. Okay, dahil mayroon atang problem sa mic si Miss, mabalad tinak na lang daw elongation. So, tingnan po natin kung tama. So, tama. May tama ka. Next po. So, the next step is elongation. Elongation is the addition of nucleotides to the mRNA strand. RNA polymerase reads the unwound DNA strand and builds the mRNA molecule. Using complementary base pairs, there is a brief time during this process when the newly formed RNA is bound to unwound DNA. During this process, an adenine or letter A in the DNA binds to an uracil in the RNA. So, wala na kasi dyan si thiamine, di ba? Pinalitan na siya ng uracil. So, once RNA polymerase is in position at the promoter, the next step of transcription is elongation. Basically, elongation is the stage when the RNA strands gets longer, kaya siya elongation. Thanks to the addition of the new nucleotides. During elongation, RNA polymerase walks along one strand of the DNA, known as the template strand, in the 3' to the 5' direction. For each nucleotide in the template, RNA polymerase adds a matching or complementary RNA nucleotide to the 3' end of the RNA strand. The RNA transcript is nearly identical to the non-template or the coding strand. However, RNA strands have the base uracil in place of thiamine, as well as slightly different to each sugar in the nucleotide. So, as we can see in the diagram po, napalitan na po yung T, diba, ng U. So, next po. Okay, sa next step ng transcription, sino pong gustong sumagot? Pa-type na lang ng ako na lang. Okay. Nauna si... Sino nauna? Nauna si Mariel. Okay, Mariel. Open your mic, please.
Rinig po ba?
Yes po.
Yes po. Ano po? Termination.
Okay, tingnan natin kung tama. Pa-next po. So, tama. Termination. Pa-next. Yan. So, termination is the ending of transcription and occurs when RNA polymerase crosses a stop or termination sequence in the gene. The mRNA strand is complete and it detaches from DNA. Once a gene is transcribed, the prokaryotic polymerase needs to be instructed to dissociate from the DNA templates and liberate the newly made mRNA. Depending on the gene being transcribed, there are two kinds of termination signals, but both involve repeated nucleotide sequence in the DNA template that results in RNA polymerase stalling, leaving the DNA template and freeing the mRNA transcript. On termination, the process of transcription is complete. In a prokaryotic cell, by the time termination occurs, the transcript would already have been used partially synthesized numerous copies of encoded protein because this process can occur concurrently using the multiple ribosomes or polyribosomes. So, there are two major termination strategies found in bacteria. It is the row-dependent and row-independent. In row-dependent termination, the RNA contains a binding site for a protein called row factor. So nakikita natin dyan yung row factor, yung parang string na nakababa. Row factor binds to this sequence and start climbing up the transcript towards the RNA polymerase. When it catches up the polymerase at the transcription bubble, row pulls the RNA transcript and the template DNA strand apart, releasing the RNA molecule and ending transcription. Other sequence found later in the DNA called the transcription stop point cause RNA polymerase to pause and thus help row to catch up. The next one is row independent termination. Next po. Yan. Row independent termination depends on specific sequence in the DNA template strand. As the RNA polymerase approaches the end of the gene being transcribed, it hits a region rich in C and G nucleotides. The RNA transcribed from this region folds back on itself and the complementary C and G nucleotides bind together. The result is stable hairpin that causes the polymerase to stall. In a terminator, the hairpin is followed by a stretch of U nucleotides in the RNA, which match up with the nucleotides in the template DNA. The complementary U and A region of the RNA transcripts forms only a weak interaction with the template DNA. This coupled, the stalled polymerase produce enough instability for the enzyme to fall for and liberate the new RNA transcript. So after termination transcription is finished, an RNA transcript that is ready to be used in translation is called a messenger RNA. In bacteria, RNA transcripts are ready to be translated right after transcription. In fact, they are actually ready a little sooner than that. Translation may start while transcription is still going on. So, pa-next po. Pa-next po. Yan. So, review and summarization. Transcription has three main steps, which are initiation, elongation, and termination. Laging tatandaan during the initiation, the nucleus, it is happen in the nucleus. RNA polymerase recognizes recognition sites, causing it to bind to the promoter or the start of the gene. The RNA polymerase then separates the DNA into single strand so the template strand can be read in the 3' to 5' direction. The next is the elongation. Pre-mRNA nucleotides are quickly paired with their complementary bases, which corresponds with the template strand of the DNA. The pre-mRNA moves in the 5' to 3' direction, while the template strand of the DNA moves oppositely from the 3 to 5' direction. Pre-mRNA does not contain thymine. Instead, uracil is used as the complementary base for adenine. The last is termination. When the RNA polymerase reaches the terminator, it signals the RNA polymerase to stop and release from the DNA. Once separated, the two DNA strands come back together and reform the double helix. The newly formed pre-mRNA molecule is then now released. So, next po. Yan. So, before I gave the floor to the next presenter, I have here one question for you guys. If meron ba talaga kayong nag-get sa mga pinagkukuda ko, pwede ba ako magtanong? Or baka ayaw nyo? Okay lang naman. Ito yung... baka kasi ayaw nyo. So, ito yung tanong ko. What is a promoter? It is... Is it located at the upstream or downstream end of transcription unit? So, Mr. Regacho, nakita ko, kanina pa siya nagko-comment kasi di ko natatawag. Kasi nahuhulay. So, ano po sa...
Yes po. Based on the discussion po, pag sinabi po natin promoter, it is the region of DNA to which RNA polymerase binds to begin a transcription. And to answer the question where it is located, promoter located at the upstream end of the gene, especially in transcription unit po. Ngayon lang, thank you po.
Ayan, so thank you. Wala na akong madadagdag dahil sinagot niya na tama. Napakagaling. So nakikinig talaga kayo. Maraming salamat sa inyo. So, Miss Talia, you may now start your presentation. Thank you.
Yan, thank you, Miss Suarez. So, good day good day everyone. I'm Janica and Talia, so let's move on to eukaryotic RNA processing. Next slide po. So in this process, we're modifying our pre-mRNA to make mRNA. So when we say pre-mRNA, it is a primary transcript or ito yung mga initial form of messenger RNA in eukaryotes. So ito yung mga immature mRNA na need mag-undergoes in several processing inside the nucleus. So once na-process na siya and ready to be exported na from the nucleus, pwede na siya isend into the cytoplasm for the translation. So, si pre-mRNA, hindi siya agad-agad pwede i-move into the cytoplasm without being processed or without being mature transcript, lalo na kapag eukaryote cells. Bakit kaya, no? Because pre-mRNA is vulnerable to breakdown by RNA degrading enzymes. So, need niya muna ma-process and need niya muna mag-stabilize and become mature transcripts. So, once na maging mRNA na siya or mature transcript, it can be used to guide for protein synthesis in the cytoplasm. So, next slide po. So this diagram shows the gene expression summary in eukaryote. So we're done with the transcription. So we will focus on pre-mRNA process. So as you can see, pre-mRNA process, it is also happen inside the nucleus. So it has several processes, the copying, polyadenylation, and splicing. So we will discuss it in more detail in the future lecture. Next slide po. And the newly transcribed eukaryotic mRNAs must undergo several processing steps before they can be transferred from the nucleus to the cytoplasm and translated into a protein. So these are the three main processing steps: cutting at the 5' end, addition of poly-A tail at the 3' end, and lastly is the splicing to remove the introns. Next slide po. So let's begin with the capping at the 5' end or in addition of the 5' end guanosine triphosphate cap. So after termination in eukaryotes, the end product is going to be a pre-mRNA. So before pre-mRNA actually leaves the nucleus, the 5' end of the pre-mRNA is altered by the attachment of guanosine nucleotide by the special type of linkage known as 5' triphosphate linkage. So let's pretend that this diagram on your screen is the 5' end of the pre-mRNA. So what actually we do is we add this guanosine nucleotide by the via triphosphate bond. So we called it triphosphate, but because it has 3 phosphorus atoms, as you can see on your screen, diba? Yung naka-square. And it is called 5' triphosphate linkage because that linkage is between the 5th carbon of this ribose sugar. So, ayan, bakit nga ba may capping process tayo, no? Why we should add this cap to our pre-mRNA? Is it because the important function of this cap is to protect pre-mRNA from the degradation, as I said earlier, di ba? Pwede siya ma-degrade or mag-breakdown, no? Once na hindi siya ma-process. So merong degradation that could take place during the translation or during the protein synthesis. And the addition of 5' also helps the pre-mRNA to leave the nucleus, and it also helps the pre-mRNA to stabilize and move to the cytoplasm. So let's move on to the second type of process. Next slide po. Ayan, known as the polyadenylation or the addition of poly-A tail at the 3' end. Ayan. So before the pre-mRNA actually leaves the nucleus, the tail has to actually be removed. So a small section of the tail of the pre-mRNA is removed, and instead we add many adenosine nucleotide, this 3' tail, now known as poly-A tail. So ayan, at the 3' end of pre-mRNA molecule, so mag-a-add tayo ng poly-A tail. So ilang poly-A tail bang i-add natin? So syempre, 200 to 250 adenosine nucleotide ang need natin. So, gaya ni 5' this process also gives pre-mRNA ability to resist the different type of degradation in the cytoplasm and gives our mRNA stability. So, ayan, next slide po. Ayan, so let's move on to the final process. This is known as the splicing. So in this process, we remove the regions known as introns and we combine the regions known as exons. So syempre, hindi lahat ng regions of pre-mRNA molecule ay pwedeng ipang-code for our proteins. So kaya meron tayong tinatawag na splicing kung saan nire-remove si introns. So bakit ba nire-remove si introns? Because these regions do not code for protein. Kaya nga tinawag siya, di ba, non-coding regions, si introns. So, ayan. So, meron tayong special type of enzymes yan na nagre-remove ng introns while combining together the exons. So, this is known as the spliceosome. So, as you can see on your screen, meron tayong diagram dyan ng pre-mRNA molecule. So, si exon, ayun yung may red na parang line dyan. And then, yung medyo pinkish, yung intron. Another is the red. So, alternate sa exon 1 intron and exon 2. Now, si spliceosome ay, siya yung magagalaw dyan. Siya yung magka-cut off kay intron habang kinocombine niya yung dalawang exon. So, ang magiging result niyan ay yung nasa baba. Ay yung, ayun, may line sa baba. Yung exon 1 and exon 2, ayan, magkadikit na sila. So, ayun yung tatawag nating mature transcript. O ito na yung, ayun na, mature transcript, o ito na yung mismo mRNA na pwede na mag-move into the cytoplasm na pwedeng gamitin for protein synthesis and translation. The next slide po. Ayun. Ay, last na ba? So, ayan. Question po. What would happen if the 5' methyl guanosine and folic A tail were not added to the to an mRNA? May I call Miss Han? Miss Mendoza. And sabi ni Miss Han, the transcript will degrade as the mRNA move from the nucleus to the cytoplasm, and it would not be stable enough to begin the translation process. So, tama yung sinabi ni Miss Han. So, kaya meron tayong post-transcriptional modification, or ito yung natawag nating pre-mRNA process to resist the degradation and gives pre-mRNA ability to move from the nucleus and to move into the cytoplasm for the translation. So yun, that's all on my part. May I call Miss Angel Soriano to discuss her part. Thank you po.
So, thank you for that, Ma'am Janica. So, andyan pa ba kayo, 3B? So, kindly put an emoji on our in-call messages kung talagang nandyan pa kayo. Any emoji po will do. So, ayan. Parang may mga umiiyak na, yung iba, nagugulat, iba, ano to? In love? In love ka ba, Ben? Ikaw rin, Ma'am Gelig. So, ayan. Thank you po sa mga response nyo. Talagang nandyan pa kayo. So, again, good day, everyone. I am Angel Soriano to discuss the protein synthesis, or also known as translation. So after mRNA is synthesized at the nucleus, it will now go on to the cytoplasm via nuclear pores, and it will bind to a ribosome. Then the process of protein synthesis will begin. So translation is the second part of gene expression. Dahil yung una is yung transcription, which is yung diniscuss ni Ma'am Erica. So in the process of translation, it involves the decoding by ribosome of mRNA message into a polypeptide product, which is yung protein. So based on the picture you see on your screen, what did you observe? Kindly utilize our chat box po. So we have here bacterial and eukaryotic. Sabi ni Ma'am Eunicia. Ito sabi ni Ma'am Abalod, central dogma. So all of your answers are correct. So yung dalawang picture na yan, of course, yung nasa left side is a bacterial cell, while yung nasa right side is eukaryotic cell. So you will notice that the transcription and translation happens in cytoplasm in a bacterial cell, while the transcription and RNA processing occurs at the nucleus in a eukaryotic cell. However, the translation happens at cytoplasm, particularly at a ribosome. So thank you for your answers. So the protein synthesis machinery. So basically, the machinery talks about the components needed to build up proteins. So these are the following components. So translation, the cast of characters. So first, ribosomes. So ribosomes carries out the process of polypeptide synthesis. Next is the tRNA molecules. It aligns amino acids in the correct order along the mRNA template. Third is amino acid tRNA synthetase. It attaches amino acids to their appropriate tRNA molecules. Fourth, mRNA molecules, which encodes the amino acid sequence for the polypeptides being synthesized. And lastly, protein factors, which facilitate several steps in the translation process. So guys, to easily understand or master this concept, kindly remember this in this way. So ribosome, it carries out. tRNA, it aligns. Amino acid tRNA, it attach. mRNA, it encodes. And lastly, protein factors, it facilitates. So introducing this cast of characters, let's begin with ribosomes. So ribosomes. Ribosomes play a central role in this process dahil dito nga nagaganap yung protein synthesis. Yung ribosome, ino-orient niya yung mRNA at yung amino acid carrying tRNAs para yung genetic code ay mabasa ng maayos at makatalyze ang peptide bond formation to link amino acid into a polypeptide. So, ribosomes consist of large subunit and small subunit. So, as you see on your screen, this is an example of ribosome. So, large subunit, ito yung blue. Nakikita nyo, ayan. Yung small subunit is itong yellow. So, both of them consist of proteins and one more ribosomal RNAs, or also known as rRNAs. So, remember that ribosomes are located in cytoplasms in eukaryotes, in prokaryotes, while in eukaryotes, it located at the cytoplasm and endoplasmic reticulum. Again, ribosomes. So from what I've said earlier, it has a large subunit and a small subunit. The small subunit is responsible for binding mRNA template, and the large subunit is responsible to bind tRNAs. So ribosomes have 4 important sites. So the mRNA binding sites, ito yon. The amino acid or A site, which binds each newly arriving tRNA with its attached amino acid. Next is yung peptidyl site, which is also known as P site, where the tRNA carrying the growing polypeptide chain resides. Lastly is the exit site, or also known as E site, which from the tRNAs leave the ribosome after they have discharged their amino acids. So later on, you will see how those subunit works. So, tRNA. In the process of translation, a cell reads a genetic message, which is yung nanggaling nga sa mRNA, and builds a polypeptide accordingly. The message is a series of codons along an mRNA molecule, and the translator is called transfer RNA, or also known as tRNA. So the function of tRNA is to transfer an amino acid from cytoplasmic pool of amino acid to a growing polypeptide in a ribosome. tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long, comparably to the mRNA, which is about hundreds of nucleotides. So the presence of complementary stretches of nucleotide bases that can hydrogen bond to each other allow this single strand to fold back on itself and form molecule with a particular 3D structure. Flattened into one plane to clarify this base pairing, a tRNA molecule looks like a clover leaf, gaya ng nakikita nyo sa inyong screen. So next, the 3' end, saan ba yun? Ito, it acts an attachment site for amino acid. The loop extending from the other end or the L site includes the anticodon. Anticodon is the particular nucleotide triplet that base pairs to a specific mRNA codon. Thus, the structure of tRNA molecules fits its function. Anticodons are conventionally written as 3' to 5' to align properly with codons written in 5' to 3' because RNA strand must be anti-parallel like DNA. So let's take an example on the next slide. So ayan. So tingin sa screen. So may nakikita kayong binilugan ko diyan. So let us consider this mRNA codon 5' GGC to 3'. So based on the picture, anong anticodon ang magpe-pair up sa GGC codon na ito? So you can utilize the in-call messages po. Based on the picture, ano sa tingin nyo yung magpe-pair up na anticodon sa codon na GGC? Ayan. So thank you for your responses. Tama po. So yung anticodon na magpe-pair up sa codon na GGC is yung CCG. Very good. So, habang nagmo-move yung mRNA through ribosome, a particular amino acid will be added, just like dito. So, kapag na-add na po yung anticodon, nag-pair up na po yung anticodon and yung codon, magli-leave ng amino acid which is in GLY, which is stand for glycine. So, and codon by codon, the genetic message is translated and added to a growing polypeptide chain. And from our previous discussions, it was already discussed that the polypeptide chain consists of a chain of amino acids, and amino acids are the building blocks of protein. Tama po ba? So, yan. Next po natin. So, ayan. The amino acid tRNA synthetase. So, nagtataka kayo kung bakit palaging tama yung mapoproduce na anticodon sa codon na nasa mRNA molecules. So, dahil dito yun sa amino acid tRNA synthetase. So, amino acid synthetase is an enzyme that attaches the appropriate amino acid onto its corresponding tRNA. So the active site for each type of amino acid tRNA synthetase fits only specific combination of amino acid and tRNA. And take note that there are only 20 different synthetase, one for each amino acid. And synthetase joins a given amino acid to an appropriate tRNA. One synthetase is able to bind all the different tRNAs for its particular amino acid. The synthetase catalyzes the covalent attachment for the amino acid to its tRNA in a process driven by hydrolysis of ATP. The resulting amino acid tRNA is also called charged tRNA. So next. So punta tayo sa the genetic code. Alam kong familiar na kayo since na-discuss na to ni Sir Regatio kanina. So at this point, the cellular process of transcription generates mRNA, a mobile molecular copy of one or more genes with an alphabet A, C, G, and U. So each amino acid is defined by a 3-nucleotide sequence called triplet codon. The relationship between a nucleotide codon and its corresponding amino acid is called genetic code. So ito yon. Tawag dito is genetic code. So the question is, how do we read this codon? How do we read this codon chart? So as shown in the figure, the codon AUG codes for the amino acid methionine. Ito yon yung yung AUG methionine. So this codon is a start codon. It begins the translation. Tatandaan niyo yan. Yung translation, mag-i-start siya kapag nagkaroon na ng codon na AUG. So, the start codon establishes the reading frame of mRNA. So, what is the reading frame, by the way? So, reading frame is the way the letters are divided into codons. After AUG start codon, the next three letters are read as the second codon, third, fourth, and so on. So the mRNA molecule is read codon by codon until it reaches the stop codon. So ano yung mga stop codon na nakikita nyo sa inyong screen? So there are only 3 stop codons we have. So what it is? So paki-input lang po sa ating in-call messages. So ayan, bibilis din po nilang sumagot. UAA, UAG, UGA. So very good. Tama po 'yon. So ito yung ating mga stop codon. Mamaya makikita din natin kung paano nga ba nagpa-process yung translation mula sa methionine na start codon na mag-e-end dito sa stop codon na UAA, UAG, and UGA. So next po. So, try natin yung galing nyo ngayon. So, what are the amino acids from mRNA template UAG, ACU, and AAU? So, I'll give you 30 seconds to answer this. Kindly put your answer on our in-call messages. So, ayan, may nakikita na akong sumasagot. So, ang sabi po nila, MET, THR, ESR. Sabi po ni Ma'am Eunicia, methionine, threonine, asparagine. So, let us see kung tama po yung mga sagot nila. Charan! Ayan, MET, THR, and ASN. So, very good kayo dyan, guys. So, AUG, ito, methionine. ACU, saan ba yun? ACU. THR or threonine, and AAU, which is ASN. Ito. So, ang gagaling nyo na, guys. Pwede na kayong graduate. So, ayan. So, papunta pa lang tayo sa exciting part dahil wala pa tayo sa pinaka-process ng translation. So, ayan. The mechanism of protein synthesis. So, the first step in translation, the initiation. So again, the initiation occurs when the mRNA binds to a ribosome, particularly at the small subunits. And then, a specific initiator, tRNA, which carries amino acids methionine, will start the translation. So gaya nga nang nasabi ko sa inyo kanina, lagi magsisimula ang initiation with a particular amino acid methionine or the AUG. Ito yung ating start codon. So, in either bacteria or eukaryotic cells, always remember that AUG, methionine, signals the start of translation. So, the presence of methionine is important. So, why? So, it is important because it establishes the codon reading frame for the mRNA. So, hindi naman pwede mag-continue yung mga ibang codon kung wala yung start codon. Tama po ba? So, the attachment of mRNA, the initiator tRNA, and the small ribosomal subunit is followed by the attachment of large subunit. Ito yun. So the attachment of large subunit will complete the translation initiation complex. So at the completion of this process, the initiator tRNA sits in the P site, ito yung P site kanina sa gitna, and the vacant A site, ito, is ready for the next amino acid tRNA. And also remember that a polypeptide is always synthesized in one direction, from the initial methionine at the amino acid, which is called N-terminus, while the towards the final amino acid at the carboxyl end is called C-terminus. Next process is the elongation. So kindly paki-zoom na lang yung ating chart. So next is yung elongation. So after the initiation, of course, we are now on elongation. In the elongation stage of translation, amino acids are added one by one to the previous amino acids at the C-terminus of the growing chain. Each addition involves several proteins called elongation factors. It includes 3-step cycles. So the energy expenditure occurs in the first and third step. So the first step is the codon recognition. Codon recognition requires hydrolysis of one molecule of GTP, which increases the accuracy and efficiency of this step. And then one more GTP is hydrolyzed. One GTP is hydrolyzed or broken up to provide energy at the translocation step. The mRNA is moved through a ribosome in one direction only, from 5' and first, and this is equivalent to a ribosome moving from 5' to 3' of a mRNA. So the main point of this ribosome and the mRNA move relative to each other is what we call unidirectional, codon by codon. The elongation cycle takes less than 1/10 of a second in bacteria and is repeated as each amino acid is added until the polypeptide is completed. The empty tRNA that is released from each site will return to the cytoplasm, where they will be reloaded with the appropriate amino acid. So the last step is the termination of translation. So this is the final stage of translation. Elongation continues until a stop codon, yung sinabi ko na sa inyo kanina, reaches the A site. The nucleotide base triplets, the UAG, UAA, and UGA, which is all written by 5' to 3', do not decode for amino acid but instead act as a signal to stop translation. So, as a release factor, a protein shaped like amino acid tRNA binds directly to the stop codon in the A site. The release factor causes the addition of water molecule instead of an amino acid to a polypeptide chain. The bond between the completed polypeptide and the tRNA in P site, releasing the polypeptide through an exit tunnel of the ribosome large subunit. The remainder of the translation assembly then comes part in a multi-step process, aided by other protein factors. They broke down the translation assembly, which requires hydrolysis of 2 or more GTP molecules. So, ayun na yung end ng translation. So, summary ng buong central dogma. So, this diagram shows the path from one gene to one polypeptide in eukaryotic cell. Each gene in DNA can be transcribed repeatedly into many identical RNA molecules, and each mRNA can be translated repeatedly to yield many identical polypeptide molecules. And take note that the final products of some genes are not polypeptides, but instead RNA molecules that do not get translated, including tRNAs and rRNAs. So overall, the steps of transcription and translation are similar in bacterial, archaeal, and eukaryotic cells. The major difference is that the occurrence of RNA processing in eukaryotic is located at the nucleus. Other significant differences are found in the initiation stages of both transcription and translation, and in the termination of transcription. So, para mas maintindihan nyo po yung naging process ng central dogma, panoorin natin to.
In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So ayan, nakita niyo na po yung process ng central dogma. Punta na tayo dito sa... Saan na yun? Yan, sa last part. May tanong ako sa inyo at ito yon. What two processes ensure that the correct amino acid is added to a growing polypeptide chain? So anyone from the cohort? So ayan, si Miss Mariel Lontayaw. Ma'am?
Hello. Naririnig po ba?
Yes po.
So, yung two processes po is, yung una, kada amino acyl tRNA synthetase specifically recognizes a single amino acid. Tapos po, taga-attach po siya sa may appropriate tRNA. And then, yung pangalawang process naman po, yung tRNA charged with its specific amino acids. Tapos, nagbabind lang siya sa mRNA codon. So, yun lang po. Thank you.
Ayun, halatang nakikinig talaga si Ma'am Mariel. Very good ka, ma'am. So, thank you for listening. So, let me call again Ma'am Miriam Tarega to discuss our teaching strategies. Thank you.
Ayan, so nandito na talaga tayo sa ending part. Ito, so, let's proceed to the teaching strategies. Next po. Ayan, first is, finally, oo nga eh. So, finally, first pala yun, nadadala ko dun kasi excited na rin ako. First is integration of technology. Next po. Ayan, so, the integration of technology in teaching science concepts allows full engagement and participation of the students. It tickles the interest and imagination of the ideas connectedly in the topic. And lastly, it lets the students to see the reality or even the representational images of science concepts. So, through the integration of technology, napapalawak natin yung imaginations, and also, may mga bagay kasi na sa science, di ba, hindi natin na ipapakita lively yung mga concept, which is napapakita siya through video and other types of concept showing in ICT na kapag shinow natin is makikita nila and may imagine nila kung ano talaga yun. So, next po. Inquiry-based instructions. Next. Yan, we have 4 phases of inquiry-based learning. Una is interaction, pangalawa is clarification, questioning, and then design. Sa interaction naman, it is dived into engaging relevant and credible media forms to identify a need or opportunity for inquiry. So dito, ano siya, open-minded, na critical yung pagiging curious ng students. So next is yung clarification. Dito naman is by summarizing and categorizing data and understanding with teacher or expert support. So dito naman is more on reflective yung questions. Next is questioning, asking relevant questions to drive continued self-directed inquiry. So dito, independent yung way ng questions and also creative. Next naman is design, designing an accessible, relevant, and curiosity-driven action or product to justify inquiry. So, as well as the questioning, creative din yung type ng questioning dito, and restrained. So, next po. Yan, effective questioning. Next po. Yan. Meron tayong 4 na questioning strategies for effective and thoughtful teaching. Una is yung agree, build, challenge. Ayan yung part ng teaching na, part ng questioning strategy na, i-ask natin kung agree ba yung mga students sa sinabi natin, or kung hindi, bakit, at i-justify nila yung mga sagot nila. So, through that, na enhance natin yung higher order thinking skills nila. Next is yung cold calling. Cold calling naman is it is making the students always stay alert kasi ito yung parang warning na anytime tatawagin kita. So, yung mga students na yan, students natin is always magiging attentive sila. So, doon, makikinig sila all throughout the discussion. So, next naman is yung wait time. Ito yung waiting for the questions to sink in before calling on a student to answer. So, syempre, magbibigay muna tayo ng time after natin ibitaw yung question para bigyan sila ng panahon mag-isip. And then, saka natin sila tatawagin. So, last naman is yung no calling out. Ano ba yung calling out? Ito yung, yung, sorry guys mahina na yung sounds kasi ang lakas ng ulan. I mean, sa no calling out, ito yung madalas na nangyayari, lalo na sa junior high school or elementary schools, which is yung mga students ay bine-blurt out nila yung answers nila. So, dapat hindi natin yun ito-tolerate. Halimbawa, may nangyaring ganon, hindi natin i-call out kasi bakit? Ano nangyayari? Pagka ganon kasi, hindi nahihinto yung others na mag-isip kung ano pa ba yung ng answer sa question na binigay natin. Parang tinolerate natin yung pag-blurt out ng answer. So, yung iba din na mag-iisip, okay, nasagot na to, okay na to, ganun. So, ayun lang. Next po. Yan, unlocking difficulties. Ayun yung ginawa ni Miss Suwal kanina. Next po. Ayan, yung it allows students to understand the meaning of complex words before encountering them. And because it broadened their comprehension of increasingly complex words. So, pinapresent muna before the lesson yung mga difficult words na maaaring ma-encounter para una pa lang alam na nila kung ano ibig sabihin. So, next. Learning by doing. Next po. Ayan, it allows students to fully grasp the topic since they perform themselves and discover many concepts aside from the lecture. And performing experiments, exploring simulations, role-playing are examples. Ayun. So, yun. Through experiments by exploring simulations, doon na de-develop nila yung skills, hindi lang yung knowledge nila. And also, nasa-satisfy yung curiosity nila and na-answer nila yung mga questions nila based on sa concept na pinag-aaralan nila. So, next po. Ayan, encourage them to ask questions. Next po. Ayan, so it helps students to rationalize concepts and clear out the questions in their mind. Allow students to ask for more clarification on a certain topic so that they could understand further. So, ayan, it is very effective po na i-encourage natin sila na magtanong kasi since science tayo and also applicable din naman to sa ibang subject. Kasi yung mga bata, marami talaga silang tanong. Minsan nahihiya lang sila. Ayun, so encourage natin sila magtanong para masagot natin yung mga gulo sa kanilang mga isip. So, next po. Ayun, tapos na finally. Thank you po, thank you sa lahat sa pag-cooperate and yun. Thank you.